So here's the base camp here on the beach with a bunch of tents. Like I said, it was a pretty large operation. We did have generators, so we had electricity, so we could use our laptops, charge our cameras, use GIS equipment. So the main camp I was at, though, was a bit more remote, about a 7-hour hike from that base camp. Basically, this camp is in a volcanic crater, so Bioko is a volcanic island, and it would be a 7-hour hike up the mountain, up the volcano, and then down into the crater. So what you're seeing here is a view from inside the crater. If you were to pan 360 degrees around, you would see the crater walls all around you. So this is an area where the monkeys were most abundant. The volcano is inactive, at least that's what I've been told. So this is a picture of me organizing my samples at night. This is Jake Owen, a grad student from Drexel University who's studying primate feeding ecology on the island. He's picking up a nice little present that a primate has freshly left him. We got some really good pictures of the primates on this last trip. Some of the best pictures that exist for some of these species. National Geographic came to Bioko a couple years ago and did a full spread on the Bioko monkeys, and they still didn't get pictures that were as nice as some of these. This is probably the best picture we have in existence for a Preuss's monkey, and this is a red-eared monkey. So these are both very endangered species that only exist in this part of Africa. The other two places I've been, I want to talk about together because they're similar experiences but very different than what I've been talking about. One is, I mentioned earlier, Kibale National Park in Uganda, and the other is the Adzungwa Mountains in Tanzania. And the reason why these are similar and different from the other experiences is because there are biological field stations there where people have been conducting long-term research. In some cases, for example, at Kibale, there have been decades and decades of research being conducted on the primates there. So I want to focus on the Adzungwa Mountains, though, because that was my more recent trip. I was in Kibale in 2006. I was in the Adzungwas last summer in 2009. So these are the primates that you find in the Adzungwa Mountains. It's a very diverse community. You have the Rungwacebus kapunji, this recently described monkey that's critically endangered. You have the Sanjay mangabe and the Adzungwa red colobus, both of which are only found in this area and both of which are endangered. And you have these other primates as well. So the main problem in the Adzungwa Mountains is habitat fragmentation. So you have these large forest blocks that are separated. It's hard to tell with the scale of this map, but these forest blocks are very large, and the distance between them is very large as well. So primates cannot cross these areas where the fragments are separated, and these fragments are maintained by human activity. So, so over the course of time, it's been humans that have been fragmenting these forests. So this is the research station where I was based. So you can see there's a satellite here. So you have satellite for internet. There's running water. There's electricity. Food options aren't so great. There's not a lot of protein there. So hard-boiled eggs and rice and beans just about every day for every meal for about 3 weeks. Got pretty old, but it is what it is. This is the house I stayed at. And these are student dormitories that are being constructed. They should be done by now. They're supposed to be done by December. So they're really building up their infrastructure for research and education here. So hopefully, not this summer, but maybe summer 2011, I'll be able to teach a field course here in primate conservation biology. If you want to take that, whether you're a student here or not, we can talk about that. This is the view of the forest from the field station. So this is a mountain forest, which is really interesting. As you go up the mountain, the tree species composition really changes. So that allows for a change in the animal species as well. So you have different levels of abundance of these primate species at the base of the mountain than what you see at the top of the mountain. So a lot of interesting gradients going on that people can study. So that's the view from the field station. So if you go to the top of the mountain, this is the view from the opposite side. So here's the field station there. So there's the field station. There are the 3 houses. The student dormitories are somewhere over here. So to give you an idea of the 2 opposite views. This is Mokoro, who was my field assistant there. Mokoro was very good at finding primates and identifying their feces, which was good because this was my first time there, so I was not really familiar with the primate species there and what their feces looked like. So Mokoro greatly helped my research. Would not have been able to collect as many samples as I did without him. And here are a couple of the pictures that we have that were taken this past trip. Here's the Adzungwa red colobus and the Sanjay mangabe, again, both endangered and only found in the Adzungwa Mountains. So these are, again, some of the best pictures I think we have of these species now. So, like I was saying, I couldn't visit all these places on my own. You know, the red stars are places I've been, but the black stars are places where people gave me samples. They were doing research there. They happened to collect samples. They sent them to me. I also utilized field and museum specimens. So, basically anything I could squeeze DNA out of, I would use. A lot of these populations are in areas of civil unrest, so it's impossible to collect from them, or the populations are so endangered, you just don't see them anymore, so you can't collect from them. So from populations like that, I had to resort to these alternative methods for DNA collection. Using all those samples, I constructed this phylogenetic tree using mitochondrial DNA, if you're interested in that information. And this is the tree that comes out. From these data, you can infer that the red colobus radiation split or began around 3 million years ago, which was much further back in time than previously thought. Some previous estimates were just a few 100,000 years ago that this radiation started. So it seems like it's a much deeper radiation than previously thought, and there's a lot more genetic diversity present than previously thought. You have 7 different, what I would call phylogroups here that represent divergent lineages. And if you map these on the red colobus distribution, this is the result that you get. So just a few notes here. I grouped phylogroups D and E together because there were members from the same population that fell into these two groups. So even though they're genetically divergent, it seems that maybe there were genetically divergent lineages that have now hybridized. This probably has something to do with the complex history of the Central African rainforest, which we don't know a whole lot about. So those two are grouped together. Bouvier's red colobus over here, like I said, is probably extinct. There are only maybe 15 specimens in the world in museums that are scattered across the world. So I was not able to sample that. We don't really know what's going on with that animal. No one really wants to go look for it because it exists in the largest swamp in Africa. My graduate advisor tried to get me to go, but I said no way. So there's a couple of notes. So what you should see here, what is interesting is you have a lot of diversity. You have these 4 different phylogroups in West Africa. So previously, most of the conservation efforts for these red colobus monkeys were being focused on East Africa because they thought that's where the greatest diversity was. But it turns out there's a lot of diversity in West Africa as well. One of these phylogroups, B over here, belongs to Miss Waldron's red colobus. So that is the species that I said, or the morph that I said was probably extinct. So it seems we've already lost a large part of evolutionary history in this group, a large part of their biodiversity. So from this, what I would say is, conservation priorities have to be shifted so that we're taking account these diverse lineages in West Africa as well. I'm not saying abandon what's going on in East Africa, but we really have to shift things so that we're paying attention to these populations, especially these really kind of restricted, these populations are really restricted in geographic range and represent these lineages. So G, C, and F, I would say, are in particular need of conservation attention. So in conclusion, the initial diversification of red colobus monkeys occurred much earlier than previously thought. So they're much more diverse than previously thought. There's a lot more evolutionary distinct lineages in there. So there are numerous distinct red colobus molecular lineages that are present. And red colobus monkey conservation priorities should be assigned to distinct evolution lineages with fragmented distributions, especially these that are found in West Africa. So by doing that, we will put ourselves in the best position to prevent a loss of biodiversity within this group. So how does this affect conservation priorities, or what actions have been done because of this work? Every 2 years, IUCN, in conjunction with Conservation International, puts out the primates in peril. So this is the list of the 25 most endangered primate species, or 25 most endangered primates in the world. So this work that I just presented was instrumental in bringing the Niger Delta red colobus onto this list. So that's this thing over here. Like I said, this was just discovered recently in the 90s. It itself represents a very distinct evolutionary lineage. It's in a really restricted geographic range. It's critically endangered. It's in an area of civil unrest. The Niger Delta is where There's a lot of oil production for Nigeria, so a lot of civil unrest is there. A lot of people are getting kidnapped and so forth. So it's very hard to implement conservation strategies there. But hopefully, this will draw attention to the problems that are involved in the existence of this animal. And in general, hopefully this work will allow conservation biologists to reassess the strategies that they're employing to conserve red colobus monkeys and other primates as well, so that conservation action is more effective in preserving biodiversity. Depends where, but in general, it's really hard to explain genetics. And it's really hard to explain why I'm following around these monkeys and collecting their feces. Yeah, it's absolutely absurd to them. But in some of these areas, like in the Adzungwas, the guides are very well trained. They work for the national park, and they've had other researchers coming in doing this. So they're more familiar with it. In some of the other areas, not so much. They don't really quite understand what exactly I'm doing or why I'm doing it. I can kind of describe it to them, but in very kind of vague terms. But as far as the details, as far as the genetics, that doesn't necessarily get through. It's certainly been going on since the 90s, probably. And it's really picked up ever since. I mean, the more the methodology gets affordable, the more it's being used. So it is a very kind of growing field right now. But it's been used for at least the past 10 years or so. But yeah, it's getting to be more and more common now. Especially people have done, if you ever heard of barcoding, so people are trying to barcode. So if you have meat that's crossing international boundaries or ivory or something, you can actually barcode it and extract DNA, and then to figure out exactly where it's coming from. So a lot of like law enforcement agencies have been looking into that as a way of tracking the movement of illegal substances or illegal wildlife trade. Well, nowadays there are these kind of handy kits that you can just buy, and yeah, these kits have these reagents where the DNA gets lysed. With the fecal samples, there's some extra steps to kind of remove any kind of inhibitors that might prevent the amplification of DNA, and then there are various kind of washes and buffers you put it through into this kind of filter tube that kind of catches the DNA. So I'd like to thank my funding agencies, Primate Conservation Inc., National Science Foundation, New York Consortium in Evolutionary Primatology, these large number of collaborators that provided me with samples. This work wouldn't have been possible without them. And then these institutions and people that helped me out in the field. And I'll just leave you with a picture of the Adzungwa red colobus.